The human brain has 100 billion neurons, each neuron connected to 10,000 other neurons. Sitting on your shoulders is the most complicated object in the known universe. As a neuroscience student, I'm interested in aiding the efforts of many scientists in mapping the connectome, which is the comprehensive map of all the neural connections in the brain. But as I'm sure you can appreciate the logistical difficulty of mapping the human brain, neuroscientists are starting this century's project with smaller organisms, such as Drosophila melanogaster, the vinegar fruit fly. There are several methods for mapping neural connections that are currently being used in Drosophila, but all have drawbacks and limitations. My honors thesis project looks at improving a technique that uses a mammalian receptor called P2X2 to force specific neurons to fire. It's a type of mind control, basically. The technique goes like this. You express the P2X2 in neurons you are interested in studying. You then apply ATP, which binds to P2X2 to open the receptor. That allows calcium to enter the cell, thereby forcing that neuron to fire. Once the first neuron fires, you get a domino effect, whereby neurons that are connected to the neuron will also fire, and will cause neurons connected to them to fire as well. This technique isn't perfect. Among other things, some weaker neural connections might be missed because the P2X2 isn't sensitive enough. Another issue is that there's a time lag between application of ATP and the firing of the first neuron. My project seeks to improve this technique to address these issues by first exploring the use of a mutated form of P2X2. This mutated form has only one amino acid that is different, but when I expressed it in Xenophus oocytes and compared it to normal P2X2, I found that this small difference translated into about 15 times higher sensitivity to ATP. I'm currently testing this mutant P2X2 by expressing it in fruit fly neurons to see if its enhanced sensitivity results in significant differences in detecting neural connections and in reducing the time lag. I will also be exploring other methods of addressing those issues, such as creating a way to express the P2X2 receptor only on the neuron's dendrites and not on the other parts of the cell membrane. This will concentrate the receptors where they are needed, potentially generating more spatial specificity and a faster response. I will be conducting proof-of-principle experiments in the clock neurons of the fly brain, a network of neurons that regulate sleep and wake cycles and are of much interest to many scientists.